for students who are interested in molecular and cellular biology. That's a concentration, we call what other schools call a major, we call a concentration, and shows you a little bit about my educational background. I should say one thing about my educational background is I'm quite a generalist. That is, when I went to graduate school, I actually studied genetics. I was a geneticist. Um, I I did genetics research as a graduate student. When I switched, the next step after getting a PhD is to move on to what's called a postdoctoral fellowship. And so you do research in a different lab. And it's often really good to learn something else that you didn't learn before because it broadens your understanding of things. And I'm going to get to this in a second. I'm going to talk about this concept generalization, because that's what our brains are actually really good at, is taking different pieces of information that they store. Of course, those are memories, and using them, piecing them together in novel ways, and we call that generalization. And so to me, it was very important to switch. And so I went from genetics and genetic research to studying the brain and cognitive functions of the animal. So that's my postdoctoral studies. And it's basically what I continued as a professor in my research. Um, but what's great about that is that it left me able to do many different tasks in terms of teaching. So I could teach a class about the brain, which I love to do because that's what I study. But I could also teach a class about, basically it's about cancer or cell biology. Involves a lot of genetics because cancer is kind of a genetic disease. So I teach that as well. Um, we always say, at least in the business I'm in, that teaching is one way that, you keep things in your mind in order to be able to generalize, in order to access information. Um, Now, in terms of research, not all my research, but some of my research has been with fruit flies, um, and specifically on the brain of fruit flies. When I was first starting out in that postdoctoral research, I was interested in the visual part of the fly's brain. And so here you see the eye. This is a scanning electron micrograph here in black and white, basically, but colorized inside. So this is just a colorized inserted picture that shows the brain of a fruit fly. Um, and there are different parts of the brain, just like different parts of a mammalian brain. There are different areas, areas involved in vision, perception, odor perception, storing memory, which I'll get to in a second. And when I started out, I was very interested in how the brain uh, forms its wiring. So basically, the connections between the neurons that make up the brain, those connections called synapses, they become established as the animal is developing. For us, it's of course in the womb during gestation, and our brains are being formed. In a fruit fly, well, it's a little more complicated because they have two different stages of their life cycle. They have an adult stage and they have a larval stage. Actually, what's really cool about this is to think that the adult fly, or this works for other insects like moths, the adult animal is actually developing inside the earlier stage animal that looks like a worm. Okay, so I was studying the visual system, and this is superimposed. I hope you can see everything, right? So somebody should tell me if you cannot see the slides. But superimposed is a fluorescent antibody staining, a fluorescent staining where we can see the photosensitive cells. So these sense light. And then in here is where that information is conducted by the axons and make connections. The connections you can see it's all very, very precise. And so during the formation, the development of this brain, there's many steps that are very intricate in order to wire this thing up. And of course, it's like your computer. If you put one wire out of place, something doesn't work quite normally as it should. Of course, I think the brain is a little more tolerant of abnormalities than, say, the computers. But um, so So I was studying this, and this is the first thing I studied in neuroscience, is how this wiring of the visual system is put together. This picture on the left shows you sort of the different kinds of neurons. And of course, if you've ever heard about something about neurons, they have these really unusual shapes for cells, right? So like long, thin axons, very complex branching dendrites, and of course, that's so that they can connect to other neurons, often other neurons that are far away from where the neuron is located. So for example, if we look up here in the eye on the outside of the animal, so this is where it's sensing light, and this is where they detect the photons of light, but that information is then brought through by the axon and down to a very specific place in this part of the brain. It's all wired up very precisely. Now, when does this actually form? Well, it forms in this larval stage animal, right? Looks nothing like the adult fruit fly. Um, but if you were to dissect this open, and I can tell you I do this by the hundreds, I pull them apart and pull out this part of the developing nervous system. You can see here, and beautiful fluorescence imaging. It shows you what our microscopes can do. Because if you think about how small this is, I mean, you of course cannot see this without the aid of a microscope. But more than that, the microscope is using fluorescence of different fluorescent dyes to be able to see different types of cells, different types of neurons. And so if I go back to this previous slide, and all of this wiring that you see here connecting different parts is actually being put together in this larval stage animal. And so this is actually up here is the eye. These are the cells that will detect the light. And these are their long axons that go down. And this is here the developing brain. Um, and you can see there are cells down here, and they're actually going to make connections, synapses with these cells down here. Now, all of this is really, you know, it's extremely small, but also very intricate and very precise in how it all is formed. One of the things I studied early in my career, of course, I've been around a long time, so I've done a lot of different things. But one of the things is a protein. And so if you're looking at these, you can see this movie, and there are little particles, they're little fluorescently tagged particles, which are actually a protein. And this protein is moving along these axons, moving along the axons and into the brain, communicating with these cells that are in here. And actually, it's really important. It's like this communication that happens between the eye and the brain during development. So anyway, this is, of course, one of the things that I was, doing way back. This is many years ago. Um, After that, I started to turn my research towards understanding how memory is formed. And so going back to this image inside, so inside the head of a fruit fly is, of course, the brain. And in the brain, there are different regions. And there's this region right here. You can see it's symmetrical. The brain is just like ours, it's symmetrical. Um, and this region shown here in green is used for storing memories. So that's basically their equivalent of our hippocampus. If you've heard of the hippocampus, they have something like that. And, in principle, in terms of like the molecular mechanisms, the synapses, a lot of the same things happen in the brain of a fruit fly. I mean, you've heard of things like dopamine, and dopamine is involved in this, um, just as it is in, say, a mouse or a human. So it's quite interesting, but also quite accessible, easy to study, of course, quite small. So, for example, one of the things we've done in research is to try to find the neurons that are storing specific memories. And we actually take a live fruit fly. So just imagine, if you can see here on the right side, and there's a student, a graduate student here, looking actually the data, he's looking through the microscope, object eyepieces, and looking down below. And what he sees is shown right here, which is a live fruit fly, which has wires attached to its legs. And this fruit fly is in the process of learning. It's going to get exposed to an odor, and then there's going to be an electric shock, and it's in the process of learning, and that will cause some neurons to make certain changes, which he's trying to observe. Now, he doesn't actually collect the data by looking through the microscope. It's done by computer, but this is probably just to find the specimen. Um, so I want to, I introduced you to all of this because basically I wanted to give you the sense that maybe, there's a lot of interesting things to learn, and it's exciting. You learn by doing. And part of learning really is doing things. Doing something is what cements it in your brain and causes this generalization that allows you to take the thing you learned and apply it somewhere else. Okay. And it's very important. The idea that you can learn something and apply it somewhere else not only helps you do something new, but also keeps that information in your brain, cements it in place. I'm going to show you a little bit about that. But first, I want to just, give you this sort of introduction here. Um, is, well, why would you want to start getting involved in research now when you're in high school? It doesn't, matter which grade you are, just, as soon as you can start doing research. Um, and one of the reasons is because learning to think in a scientific way is something that takes practice, right? And not to suggest anyone should become too nerdy or something, if that's what you think equivalent of scientific thinker is someone who's nerdy, but no, not necessarily. It's to be rational, to take information, to know when you don't understand something, and to understand that when you don't understand something, that's actually interesting because it's a question. There's a question, something you don't understand. So you can do that starting today, right? You don't have to wait. Um, and if you start to get ready to do research, then when you get an opportunity in high school, which you may occur, but certainly in most colleges you get into, you can find a way to get involved in research really very early, from the beginning, like the first year. Um, and maybe it's possible in your high school to get, involved in research. Now, I should just mention there's a student that I, had been teaching who decided to purchase some fish and do experiments at home. And it required buying a certain number of things, but actually it's not that hard to do interesting research on your own. Um, and really what's important about this is that when you start to see that what you're learning in school, you go this class and that class, and there's this information that is coming at you. But when you're actually using it and you have a reason to know this information because you're using it and because you're doing research, then it's a lot easier to learn the things that you need to learn. Okay, so I wanted to just talk a little bit about my ideas for teaching, and it's a little bit sort of colors the way I have organized this program that I'm going to talk about, which is going to be given online by Kyros, and then in the summer we do an on-site research laboratory program. Um, but it comes from, just to illustrate this, studies of how memory is first formed and then is generalized in the brain of a mouse. So this is a mouse brain shown in the drawing here. And of course, it's all very schematic, but the ideas are illustrated. So here's this, say the animal has learned something. Now, often with a mouse, the way it's learning something is some sort of, learning paradigm, and a very common one is called fear conditioning. So the mouse is in a cage with, say, certain objects or colors, so it can recognize the cage, maybe an odor. Um, and then it may get some sort of, what's called an unconditioned stimulus, like an electric shock. Okay, that's a very bad stimulus. Um, and it's called fear conditioning because it's an electric shock, it's painful, but they store the information, the association between that particular environment, the context, and that bad experience. They store that first in this area of the brain called the hippocampus. So it's encoded there. It's encoded just like I was talking before. There are neurons connected in a certain way, and they change their connection strength. And this is how information is stored. But we didn't need to get into the details. Thing is, when the animal learns this one thing, it's in this one cage and it has this experience, and there are certain, associated colors and shapes and maybe an odor. Um, it eventually transfers that information in a way that it can generalize that and in a way that it can recall that information in other contexts. It can remember that. And so what happens, this is actually known to happen in the mouse, is the information that is stored in the hippocampus will move to another part of the brain and, in other words, be encoded in this area called the prefrontal cortex. So here, so you can basically see this information represented by these orange spots is now been moved to this area, the prefrontal cortex. So it's stored here. Now, the animal goes about its life, has other experiences, and in some of those experiences, make it, recall the fact that it got an electric shock in certain conditions. And those new experiences form associations within the prefrontal cortex. So the memory that was initially a simple memory that was stored in the hippocampus becomes more generalized, connected to other memories here. And this information actually goes the other direction. So it goes back. So in other words, the information then is changed by using the information, say in a new context, so it becomes changed. And then it can go back to the hippocampus, transferred back, so that the original memory is now augmented by other associated memories that the mind has connected with them. So in other words, the animal now will be able to generalize. Maybe it sees an object that looks a little bit like the object in the cage in the first place when it got an electric shock. It looks a little bit like it, reminds it of that. And it's going to now have a behavior that it didn't have before the information was transferred to the prefrontal cortex and used in this generalizing. So in other words, the way I want to think about how you learn, and because I think this is true, is that first there is taking in information, there's learning, but as you use that information, you see broader implications for how it's used, and it falls into a network of other stored information. And then, so it's something different, and then you can use it in a more general way, right? You generalize. So the BioResearch program is set up in a way in which there's both learning and then using that learned information. And I'll show you how we do this. So it's set up in a way where there's three different parts. We call them tier one, tier two, and tier three. And in tier one, since especially for those of you who are not very knowledgeable about how biological things work, right? Like what are the components? How do they work together? The first class really focuses on, or tier one class focuses on the foundations. In other words, what are biological systems? How do they work? The second part, though, called tier two, which is coming up in the spring, um, teaches you about advanced technology. And so you'll learn about how we actually gather the information about biological systems? What is the interplay between them? And eventually, then we move to independent research projects. And we often combine tier two and tier three together. So you'd be working on research projects at the same time you're learning about technology. All right. So this is broken up, if you see on the right of your screen, into biological systems, what I call methodological advances, and then or approaches, and then we put them together in research projects, and we combine all of these things, learning and actually using the information, which helps you generalize and use that information again in the future when you're confronted with a new question or a new problem that you need to solve. I'm going to give you an example of this. So, for example, there's something people study nowadays, which is to make a brain. It can even be a little human brain. It's called a brain organoid. And these are actually little brains. These are actually two little eyes. They really are eyes, not normal eyes. But the brain actually, that's formed is quite small, but it has many of the features. And by features, I mean the different kinds of neurons, the different areas like a hippocampus and a cortex, things that are very similar to a normal brain, not organized quite in the normal way. But of course, there's a lot of work nowadays going into how to make these brains more and more similar. And we're gonna learn about this in the course. But let's consider, the knowledge of what a brain organoid is. But there's also the question of, well, how is it made? How is it done? And by learning this, it will be easier for you to understand and remember and to generalize what what a brain organoid actually is. So we consider, and just in the way I think about these things, is the brain organoid is the biological system. And then we have different methods for studying the brain organoid, which I'm going to talk about. But those, of course, are subjects of tier two, part of the program. So, but to give you an example, a method would be something like what we call synthetic biology. We take human stem cells, for example, and these are just simple single cells that are growing in a dish, but doing certain things. In other words, in the laboratory, certain things you can do will cause those stem cells to become first stem cells for neurons, and then those neuronal stem cells, if treated in the right way, will actually form this brain organoid. And so we're going to learn both how it is done and what it is. Okay. So that's what these things are. So, and taught in the tier one course and then in the tier two course. So let me give you an overview. And I know, we can't stop for questions now, but if there's going to be a Q&A, so just hold your questions towards, till the end. So let me tell you a little bit about the tier one course, the foundation of biological systems. And I hope maybe the syllabus is available to you online. And the topics we cover are, as shown here, understanding how DNA is replicated, how chromosomes are duplicated, how when cells divide, how is that controlled? Very important to understanding cancer, because that's out of control cell division. And this thing called the cell cycle is actually a mechanism that controls cell division. Then I was just talking about brain organoids, and so those are what are synthetic brains. But we also can make artificial, synthetic embryos. We can make synthetic mouse embryos, actually come out very similar to normal mouse embryos, and human embryos. They don't go very far in development, but up to the point where they stop, they actually look quite like the real thing. Um, and I also want to talk to you about tumors. And so another topic is tumors, because this is really interesting, to understand that cells really agree to cooperate to form an organism, right? They all do the things they're supposed to do, be part of a muscle, be part of the skin, be part of the gut, the brain, etc. And what's going on with tumors is when cells, for various reasons, all kinds of reasons, which we'll talk about, decide that they're not going to play by the rules anymore and act like they're actually their own organisms. Like the tumor is basically another organism growing within a person. And we do have presentations at the end. So if I quickly go through here, and I don't want to take up too much time with this, but so if we, for example, when there's a cell here and it's dividing, here shown in pale blue is the chromosomes, and red, very low resolution, but those are called microtubules. They are part of what pulls the chromosomes apart. Um, and so this, process of a cell dividing to give two cells is actually regulated by a biochemical mechanism, which we call the cell cycle clock. Uh, so we're going to talk about what is this mechanism? How is it discovered? What is it composed of? What are the different proteins and enzymes that are in it? How does it work? Um, when we talk about, say, real brains, synthetic brains, it's pretty much going to be sort of what I already talked about. I mentioned, the neural stem cells and how they are used to form a brain organoid. And also, like, what do people do with brain organoids? And, many people now, and of course, I mean, researchers in laboratories are trying to make, say, silicon and biological, brain interfaces where the two can talk to each other. So what you're seeing in this dish is actually a brain organoid um, that is electrically connected to to a computer, and so then it can actually interact with the computer. And I mean, I think it's an idea, to sort of use brain organoids as in some way as part of artificial intelligence. Um, and then, as I mentioned, to talk about, say, tumors and cancer and how they actually work, what's actually going on in these cells that makes them so, uh, unruly, right? Okay. So now, when we get through, and if you're interested, you don't have to take both courses, but if you take the tier one course, um, then you may be interested in taking the tier two course. And that's where we study how you go about using all of the technologies in order to study these things, these biological systems like brain organoids. So that's in the tier two course. And so, for example, if I cut open this brain organoid here and actually look at the cells, the neurons in it, and I said, see, this is the thing, is the information I said, oh, it's made of different kinds of neurons, can make something like a cortex. Well, the reason we know that is because we can tell different kinds of neurons by which proteins they are making, because different kinds of neurons make different kinds of proteins. They're different. And so the ones that are here fluorescent green um, are one kind of neuron, and then the ones that are fluorescent blue are different. And that's what's actually is in this brain. You can't see it unless we use this methodology called fluorescence microscopy. So you're going to learn to use that, what it's about, how it works. Um, and then we're going to take another technology, learn about another technology, which is to take any of these cells, even hundreds or thousands of these cells one at a time. And so we could pick out one cell here, one cell from this brain, and remove the RNA, the mRNA. So if you remember the, central dogma, the DNA of the gene is copied into an mRNA, and the mRNA then, of course, sometimes, not always, but usually makes a protein. Some RNAs don't make proteins. But if we remove the RNA from this single cell, we have methodology by which we can determine what is the nucleotide, the sequence of the RNA So we call this high throughput sequencing a very powerful technology. We can take the RNA that's in one of the cells and we can take every single RNA, well, not every single one, but most of them, take those RNAs, determine what they are, because the nucleotide sequence, right, the sequence of A and G and C and, well, it's uracil or U in RNA, but that's T in the DNA, right? If you remember, I don't know, but it doesn't matter if you learn this, we'll teach you. Um, but each of these sequences identifies which gene the RNA came from. And so we can count them up and we know how many RNA from each of the 10,000 different genes, how many RNAs are being made from each gene. Now that's characteristic. That tells us something about the identity, what that cell is, what kind of cell, because different cells not only make different proteins, like you can see it's green because there's a protein that's fluorescent green here, and there's the blue ones, right? They're different. But actually, we get the information about all the genes, how many RNA molecules are being made from each of all the genes. A very powerful technology, which you'll learn about. And because there is so much information there, we then turn to data science, right? We use computer software algorithms to say, tell how one cell is different than another cell. So I won't explain this, but definitely in the course I will talk about, how we can classify a cell as being similar, more similar, or more dissimilar from another cell, whether it doesn't matter if it's in the brain or the liver, wherever it is in the body, but we can classify it as being similar or dissimilar if it's, a distinct kind of cell based on how many of each RNA from each gene, it's that cell is making. So all of these different methods are combined. So you have a biological system here, the brain organoid. You have these different technologies that can be applied to study it. Fluorescence microscopy, synthetic biology, to use stem cells, single cell RNA sequencing, and then data science to study, what all of that information means. We can take that and apply it to whatever. That's the greatest thing about this, is that the technologies that you learn, synthetic biology, fluorescence microscopy, data science, single cell sequencing, DNA sequencing, can be as, powerfully applied to study the cells of a tumor, as much as, a brain organoid or whatever it is, many, many things, whatever your biological system can apply all of the very similar technologies again and again. This is why it's very useful that these, different methodological approaches are transferable between biological systems. So very important, aspect of how we go about doing research. So of course, then tier three consists of, one-to-one research projects where one-to-one means that I'd be working with you personally to develop a research project using some biological system that you're going to choose, a question you're trying to answer, and then the methodological approaches that you're going to apply in order to study that biological system and answer that question. Now, if you stick around and if you want to come, this is in June and July, will be, of course, then by then 2026, and you can come to Boston for the residence program of the BioResearch Academy. And, here we are. This is me here. Okay, yeah, that's me. And I usually wear this brown hat. Okay, and basically, I am working with you in this laboratory every single day for about 2 weeks, 2.5 weeks. And you see here students from a previous year who I know very well, and many of them, very get into very good colleges. Um, and and also, we really do high tech things. So you're not going to come to this program and learn simple things like how to do a PCR reaction. Well, we do PCR reaction, but we actually do, complicated technologies, but also we do real projects. We're trying to collect as much data as we can, so that we're very occupied collecting more and more data on specific projects. We have a bunch of different projects. They are real projects. They're real questions concerning things like Alzheimer's disease or epilepsy, or say cancer, various different kinds of projects that are going on. They come from laboratories in the Boston area, and we import questions and do research in those projects. These are just some of the different classes from each various different years. So just want to emphasize then, what are the takeaways from this program? Well, you gain a high level understanding of really what is cutting edge biomedical research, and it serves, I think, will serve to guide you in your future. You gain this head start on research that I've been talking about. I should mention here, number three, that I am really supportive of the students who take my courses. And that also means that you can contact, and many of the students are emailing me just recently because they're doing college applications. So, um, and there are other, benefits. So for example, um, you can really easily transition to laboratories, say, it doesn't matter where it is. I mean, but any laboratory that's doing interesting science you'll be prepared by this to get into the laboratory and actually do something. You'll understand how it works. Or if you're interested in doing things like the biology Olympiads, different kinds of competitions like Regeneron, etc., this is actually a very good first step, and even, a step that can be lengthened into multiple steps to get you along these different paths. Um, and, you can actually, use your experience in this program to help you get into other programs in the future, especially, of course, if you're starting early. So that's basically all I have to say. I just want to emphasize again, this is all about teaching you right at the beginning and to get going right away on how to think like a scientist, to be prepared to do science, so when you get to college, somebody doesn't have to explain to you what is research, right? You're going to know, and you're going to have experience, and you're going to know about technologies that they're going to be using in college, university laboratories. Um, and, finally, it's very important to realize that the greatest part about research is the fact that when you're doing research that is real research, where you're answering questions that haven't been answered, there's the chance of discovery. And it's kind of like winning the lottery. It's, that moment where you feel like you've done something so important, that nobody else has done, right? Knowledge about something, hopefully an important thing, um, that is new on planet Earth, right? You've discovered something that nobody else knows. And so instead of reading a book and having people tell you how things are, you actually are in the position to tell other people something that they don't know. And I find that to be really motivating. When you have to study something that you don't think is so interesting, but becomes a lot easier to study it when you think, oh, I need to use that technology. I need to know about that in my research, right? So any case, so that's all I have to say. And I'll turn things back to Ying Ying, but I'm going to stick around to answer your questions.